This is Professor Saroj Mishra from Department of Biochemical Engineering and Biotechnology at IIT Delhi, and today we will be addressing the topic of applications of biotechnology in agriculture. So, we will see as to how the modern techniques of biotechnology, which are predominantly driven through molecular biology, have made an immense contribution in the area of agriculture. So, in the first slide, I have the title of the slide, which is put down as Agricultural Practices Leading to Green Revolution. So, what you see is that in India and the world over, we have made large contributions in the area of agriculture, which have been known as green revolution. However, this kind of a development, which was called as green revolution, was primarily because of better agricultural practices, number one, and number two, by supplying a large amount of chemical fertilizers. However, today we are also beginning to learn as to what are the pitfalls of supplying these chemical fertilizers and how they can be very detrimental to the health of the soil. And that is where biotechnology will come in and try to address some of these problems. So, in the first slide, you see that there are three boxes which are given to you. So, the first area that we are going to look at is how biotechnological tools can be used in order to supply supplements which are predominantly the biofertilizers. And this is the point which is mentioned here in the first point, that is the chemical fertilizers which have been supplied till now, and the objective of which is to supply nitrogen, phosphate, potash into the soil. These can be replaced by now supplying what is called as biofertilizer. So, this is where we are going to be learning as to what biofertilizers are about. And the second point that you look at here is dispensing of agro residues such as straw and bran. Now, in a field, once a crop is collected, let us say it is a wheat crop, then there is a lot of straw which is left behind and which has been burnt, and you know, the lot of the problems which have been in the past recently, which is because of the immense pollution, etc., is because of burning of the straw. Now, the question is that can biotechnology be used in order to convert this straw, which is otherwise an agricultural byproduct and a waste material, or wheat bran for example, can it be converted into something which is beneficial? For instance, can it be converted into compost, which is given in the last point here. So, whether biotechnology can contribute to making a compost out of these agro residues, or the term valorization means that you want to make a value addition to compounds like straw and wheat bran. Now, as far as the wheat bran is concerned, there is not so much of excess of wheat bran which comes out of the field, because wheat bran can be used as a supplement in a lot of food material. So, you do not have a large amount of this wheat bran which is available. However, wheat straw is still a problem, and it can be converted to either compost or to some other high value product. Now, in the second box is where application of biotechnology is going to be discussed, that is in terms of either having high yielding crops or bettered crops. By bettered, what we mean is that these are your conventional crops such as wheat and rice, etc., but in addition to that they have also acquired some other good property, such as what we see in the case of golden rice, which is a rice crop, but it is able to make vitamin A also. All right. So, right here the first point illustrates that these high yielding crops have been generated so far using the conventional practices in the agriculture, such as the hybrid vigor breeding programs, where you have essentially two crops which have some very good properties. One could be that the grain is very high quality, or let us say in the case of rice, it is a very long grain, and the other could be some other good property. The weight is also very good. So, these two crops can be brought together, and then you can just bring the traits of both these crops together into a single hybrid. All right. And so, this hybrid vigor meant that you were able to combine the properties of the two crops together using conventional mating programs in the field of agriculture. Today, through genetic engineering, there are several new qualities which can be also introduced. Some of these are listed here. For example, you could have very high yielding crops, that is per hectare of land you can generate now many more tons of crop, many more tons of the grain can be generated, or the oil content of the crop can be increased, or you could make a wheat crop which is gluten free, or you could make golden rice, so on and so forth. So, several new qualities, for example, can be introduced using the genetic engineering techniques. In the third box, I will give you an example of developing a pest resistant crop. Now, till now, the pests which are generally infecting your crop plants can be removed using these conventional pesticides. However, many of these pesticides are highly toxic. So, if these are toxic, that means that eventually the soil health is going to be compromised. So, instead what through the roots of biotechnology or through the techniques of biotechnology, what can be done is that we can create crops which have this pest resistant property. For example, you have already heard of the BT cotton, which is a cotton crop, however, which is now resistant to Lepidoptera pest, and similarly BT brinjal, and these are also what are called as the transgenic crops, because here you have introduced some kind of a foreign gene has been introduced, and that is why these crops are called as the transgenic crops. Or the second route is to develop pest resistant crops is to apply the technology of silencing the RNA, which is called as the siRNA mediated technology can be used for development of pest resistance crop. So, this only gives you an overall idea as to where all biotechnology can be applied. But what we are going to do today is that we will take specific examples and we will see as to how genetic engineering can be applied. So, in the next slide, I have listed down these three topics here. So, the first part is biofertilizers. What are these and how biotechnology has made contribution in this area? The second topic which I will be covering in detail are bettered crops, as I discussed with you, and an example which we will consider is that of the golden rice, and the third example is that of the pest resistant crop, and here the example of BT cotton will be given to you. Now, before we look at the first topic, that is what biofertilizers are, in this slide there is a difference which is listed between the biofertilizers vis-a-vis the chemical fertilizers which have been conventionally used in the agricultural practices. So, I am going to I have listed down several points here which will allow you to understand as to what are the difficulties with the chemical fertilizers and how by using biofertilizers all of these shortcomings which you see in the chemical fertilizers can be overcome. So, the first point in the chemical fertilizer is listed here, that is what are chemical fertilizers? These are chemicals which are made through chemical manufacturing processes, and they start out with very expensive non-renewable raw material. So, it could be petroleum derived products etc., which are going to be used. Number two, they are going to be very expensive. However, the advantage here is that since chemical processes have been so developed over last many decades, if you want to scale up such a process, usually there is a very easy scale up because this kind of a processing has been nearly perfected. On the other hand, if you want to look at biofertilizers, these are essentially the microorganisms, that is, these are going to be the live microorganisms which are going to act as fertilizers. So, these are microorganisms which can make ammonia. So, in other words, nitrogen can be supplied in the form of ammonia. Similarly, they can supply phosphates, they can also supply potassium, for example, to crops, and they start out with inexpensive raw material. That is, these microorganisms can be cultivated on very simple kind of sugars like sucrose or glucose, which are very inexpensive. Then microorganisms are going to use these carbon sources, and a lot of this biomass is going to be developed now. So, it could be bacteria which has been grown on a large scale, and eventually the bacterial biomass is going to be harvested and it is centrifuged in order to remove all the culture medium. Then it can be dried and it can be packaged. So, now these bacteria themselves are going to act as biofertilizers. And when we say renewable, that means the kind of raw material, the kind of sugar, the kind of medium which you are going to be using for cultivation of these bacteria is very inexpensive. It is a sugar solution, or it has some kind of trace elements, it has some kind of nutrients. So, that is what we mean by renewable. That is, the medium on which these bacteria can be cultivated is readily available, and it is renewable in nature. Now, one of the disadvantages which is in this process is that the scale up is not very well established, because when we say scale up, that means you must have the ability to cultivate all of these bacteria which are capable, as we will see in the next slide, is that these bacteria are capable of taking nitrogen from the air. You can have hydrogen which is coming in, and they are able to put nitrogen and hydrogen together to synthesize ammonia, which can serve as a nitrogen source for the plant. However, in order to cultivate these bacteria on a large scale, there are quite a number of challenges. So, you could have oxygen supply in the bioreactor, maintaining sterility, or maintaining a desirable pH, maintaining a desirable nutrient concentration in order to carry out these scale up studies. That is why we say that the scale up is still not very well established. The second point is that in a chemical fertilizers, you have hydrogen for synthesizing ammonia. You require both nitrogen as well as hydrogen. So, the supply of hydrogen is going to be in the form of molecular hydrogen when it comes to chemical fertilizers. However, in the case of a biofertilizer, the microorganisms, like you see here in the second point, is that the microorganisms can use organic reductants. So, they could start out with glucose, they could start out with sucrose. So, these are organic molecules, they are carbon containing, and they are going to reduce, for example, nitrogen into ammonia. The third point is that with biofertilizers, what we are using is that enzymes which are present in these microorganisms are going to convert nitrogen and hydrogen into ammonia. So, these enzymes are called as the biological catalysts. In the chemical fertilizers, on the other hand, you have to add many of these metal oxides as catalysts. So, both nitrogen and hydrogen cannot combine. If you put these two gases together, they will not be able to combine in order to make ammonia, but rather you would require some chemical catalysts, which are written here. Like, for example, you have aluminum, you have iron, or you have molybdenum, etc. The fourth point is that these microbes, you can cultivate them at normal temperature, which is ambient temperature as well as pressure, whereas in a chemical process you would require very high temperature and pressures. So, all of these are going to be energy intensive, and adding catalysts could also leave behind traces of very toxic catalysts. So, it is again not a very environment friendly procedure, and similarly very intensive in terms of requirement of high temperature and pressure. Then biofertilizers are going to be soil friendly. Why? Well, because these are bacteria which could normally be occurring within the soil. So, you are not bringing anything extra or anything toxic to the soil. So, we say that these are going to be soil friendly. However, in the case of chemical fertilizers, when you put your potash, or when you put these ammonia fertilizers or potassium containing phosphates into soil, then eventually due to rain or other kind of agent, many of these chemical fertilizers can also wash out into the water bodies, as a result of which there is going to be pollution. Now, for instance, if you have a lot of this nitrogen which is supplied as a fertilizer to the soil, and if there is rain, then all of this extra nitrogen gets washed into the water bodies. Now, the concentration of nitrogen is going to build up to very high level in these water bodies, as a result of which you have a phenomenon which is called as algal bloom. So, lot of algae, because now you have enriched this water body with a lot of nitrogen, so algae can start to cultivate. So, you get algal bloom, which depletes oxygen, which depletes nutrients from these water bodies. So, that also causes pollution. On the other hand, the biofertilizers, for example, they are non-polluting, because they can remain in the soil. Even if they move into the water bodies, they cannot cause something like an algal bloom. Then in biofertilizer, the efficiency of fixing nitrogen and hydrogen can reach up to approximately 90%. Okay. So, these are going to be very, very efficient kind of systems. On the other hand, if we look at a chemical fertilizer or in a plant, the efficiency is only around 40 to 45%. And the last point is in terms of cost. If we look at the input cost, that is how much is it really going to take me, how much money do I need to put in order to make 1 kilogram of biofertilizer, it is only going to be 20 paisa per kilogram of biofertilizer. On the other hand, when you look at the chemical fertilizer, what you see is that it is going to cost you 6 rupees per kilogram of nitrogen and 14 rupees per kilogram of a phosphate containing material. So, you can see already the price difference is so much. So, therefore, in many of these ways, you can appreciate as to why biofertilizers should be preferred over the chemical fertilizers. So, now let us try to understand what these biofertilizers are. Okay. So, as I explained in the last slide, these biofertilizers are nothing but microbes or groups of microorganisms which are going to be collectively taken together, and they are going to be used to supplement the plants. Okay. So, what are the major fertilizers which you need to give? So, the first thing is that these are the nitrogen fixing bacteria, and they make 60% of the global market of biofertilizers. And the second is that these microorganisms are very special category of microorganisms which have the ability to solubilize the phosphate, and they also have the ability to mobilize the phosphate which is already contained in the soil. Okay. And what kind of microorganisms are these? These could be bacteria, or these could be fungi, or these could be mycorrhiza, which are again fungi, but these are very closely associated with the roots of the higher plants. Okay. So, I will demonstrate the first example here, that is nitrogen fixing using microbes. So, here is a reaction in which you have nitrogen fixation which is carried out by microbes. So, there is a list of microbes which I will explain to you in a while, which are listed in this part. But first let us look at what is the reaction that they are carrying out. So, atmospheric nitrogen combines with hydrogen, and this hydrogen is derived from various organic sources, etc. And nitrogen and hydrogen is now going to combine in order to give rise to ammonia, as a result of which the content of nitrogen in the soil increases. So, when we look at nitrogen fixation, all right, there are 3 different categories of these microorganisms which are shown in bold here. The first category, the first member is or we can say, out of the 3 categories, this is one of the 3 categories, which is the symbiotic nitrogen fixing bacteria. Now, here is an example which is given, rhizobium, all right, which belongs to the symbiotic nitrogen fixer, and this rhizobium is found in close association with the leguminous crops such as your arhar dal and all of these leguminous crops. And the reason we use a term like symbiotic nitrogen fixation is that in this kind of an arrangement, both the bacteria which are going to fix this atmospheric nitrogen as well as the plants, they mutually benefit from each other. Okay. So, the rhizobium, that is the bacterium, is going to get organic acids, and it will get so many other nutrients from the plants. And what does the plant get in return? It gets the nitrogen, it gets the ammonia. So, that is why this association is called as a symbiotic nitrogen fixation. It is a win-win situation, both from the bacterial perspective as well as the plant perspective. Another example which belongs to this category is that of blue-green algae. So, what do? They can provide nitrogen in the paddy field. They can also provide the vitamin B complex. Now, the second group of microbes which is capable of fixing nitrogen are called as the rhizosphere associated bacteria. Okay. And as the name suggests, rhizo means roots and sphere means the area which is around the roots. So, these kind of bacteria, for example, they occur in very close association with the roots of the plants. Okay. They do not particularly benefit anything from the plants, unlike the case of the symbiotic nitrogen fixers. And these bacteria, so they will take most of these things, they will take from the soil, the organic acids, the nutrients, etc. They are going to take from the soil, and they are associated with the outside soil which is present in these, which is surrounding the plant roots. So, the bacteria which are an example here are that of Acetobacter or Azospirillum, which are associated with crops like millets or sorghum. Then the third category of nitrogen fixation occurs by what are called as the free living bacteria. Okay. And as the name suggests, that they will be freely available in the soil. They do not have any kind of an association with the plant, and they are already present in the soil as free living organisms. And one example of this category is that of Azotobacter, and what you will see here, that these are associated, these bacteria are associated with many of the cereal plants or millets or flowers, for example. So, that is how the nitrogen is going to be available to the plants. So, there is no, in other words, by using these nitrogen fixing bacteria or by applying these nitrogen fixing bacteria in the soil, the plants are not dependent on any kind of a fertilizer, because then the nitrogen is going to be supplied in the form of ammonia, which is made by all of these microorganisms. Now, the second is that of phosphate. Phosphate is also a very important mineral, and it has to be supplied to the crop plants. So, what we see here is that much of the phosphate which is in the soil is in the form of an insoluble phosphate source, and that is something which the plants are not capable of utilizing it. So, what do the microbes really do? The microbes, which themselves can be called as a biofertilizer, for example. So, this microbe is capable of solubilizing the phosphate, as a result of which it becomes now in the soluble form, and then it can be very easily taken up by the plants. Okay. Now, what kind of microbes are really these? Some of these are listed here, like Pantoia, agglomerants, or Microbacterium levantiformans, or Pseudomonas putida, etc. These are highly efficient insoluble phosphate solubilizers. So, now you will understand as to what do we mean by a biofertilizer. So, in order to summarize this slide, I will say that these are special microbes which are capable of fixing up atmospheric nitrogen into ammonia, so they can supply nitrogen to the crops, or these are phosphate solubilizing bacteria which are capable of making this phosphate available so that the crop plants can take it. Now, many of these microbes cannot be just supplied like this, but rather there could be a carrier which is required for stabilizing and increasing the shelf life of the biofertilizers, because these microorganisms need to be packaged properly. So, there are some kind of materials, for example, peat or lignite powder or clay or talcum or rice bran, etc. These are, in other words, these are going to be solid matrices, and which are mixed with these microorganisms in order to increase their shelf life. Then it can be packaged and it can be supplied to the farmer. So, that is how biofertilizers are produced. Now, in order to make this concept of phosphate solubilization clear to you, I have this slide in front of you which tries to explain as to how these phosphate solubilizing microbes are capable of converting insoluble phosphate into a soluble form of phosphate. So, in this box, what we are looking at are 4 different categories of microorganisms. The blue, what you see here, for example. So, this blue one, there is an explanation which is given in this particular box here. So, this blue symbol refers to the phosphate solubilizing fungi. Similarly, the maroon circles which you see here, they refer to the phosphate solubilizing bacteria. Okay. So, the earlier category is that of fungi, however, the second category is that of bacteria. Similarly, these thin lines which you see here in blue, what are these? These are phosphate solubilizing actinomycetes. Okay. So, these are filamentous kind of bacteria which are again capable of converting or making available phosphate from the insoluble phosphate sources. Then there is a fourth category which is shown in this triangle. So, these are called as arbuscular mycorrhiza. So, it is a special category of fungi which are very closely associated in the roots of these crop plants. Okay. And their quality is that again they are able to solubilize the phosphate. So what are these groups of microorganisms doing? And these can be collectively called as phosphate solubilizing microbes. Okay. So what do? They predominantly make acids into the medium. Okay. So from the insoluble phosphate source which is in the soil, you are extracting by way of treating those rocky materials or making those insoluble salts, whatever you have phosphate salt, you make them combine with acids, and the microorganisms are capable of secreting acid into the medium. What kind of acids are these? Okay. So like carbon dioxide comes out of bacterial respiration, that can combine with water in order to give rise to carbonic acid. So this carbonic acid can now solubilize this inorganic phosphate containing material. Okay. Or many of these are going to assimilate phosphorus from the liquid. So they have kind of materials which are exuded out from the bacterial cell, and they are capable of assimilating phosphorus. Then there is another example here, that is organic acid production. So they can make, apart from carbonic acid, you can make some other kind of acids like formic acid or lactic acids, etc. All right, and they can also be used for solubilization purpose. Or what you see in the next point is chelation of cations which are bound to phosphate. Okay. Now you know phosphate is negatively charged, cations are positively charged. So there could be a lot of, in the soil there could be a lot of these cations which are capable of binding to these phosphates. However, in that form it cannot be taken up by the roots of the plants. So, but the roots, what they can do is that they can bring about chelation, that is they can just hold it together, all of these cations which are bound to phosphorus. And once that gets internalized into the plant roots, then they are capable of using it. Or you could have certain inorganic acids can be produced, like formic acid is a very good example of that. And then in addition to that, these microbes can also make a lot of these enzymes which are listed down in this box, such as alkaline acid phosphomonoesterases or these phosphatases, phytases, etc. So all of these enzymes are being produced into the extracellular medium, that is in the soil, and then they are capable of solubilizing the insoluble phosphate or hydrogen sulfide, etc. So you will see that when we talk about phosphate solubilizing microbes, there are different mechanisms which are operating in these fungi or bacteria. So now let me give you an example. We will move to the second example, where I was talking about how can you better a crop. Okay. So the example which I am going to be discussing with you is that of rice. And how are we bettering it? We are introducing the property of vitamin A biosynthesis into the crop plant. Okay. So not only can it serve as a source of, it's a food grain, but it can also give you vitamin A. All right. So now in this box I have tried to explain that what do we mean by golden rice. Now the reason it is called as golden rice is because provitamin A, all right, not the vitamin A but the provitamin A, which is called as beta carotene, is yellow in color. So if you have a rice grain which is capable of making beta carotene, then it will be yellow in color, or the term that was coined was that of the golden rice. So golden and yellow could be used synonymously. So therefore, what is golden rice? These are genetically engineered rice crops which have the ability to make beta carotene, which is actually provitamin A. Okay. So there is a cluster of genes which is going to be introduced into the rice crop, as a result of which it is going to be having the ability to make beta carotene. Okay. And then this antioxidant then converts to vitamin A, and how does it really do that is explained in the box which you see on the right. Okay. So beta carotene, for example, if a rice plant has the capability to make beta carotene up to this stage, all right, through genetic engineering techniques, then it already has enzymes like dioxygenase, enzyme like dehydrogenase, which can convert beta carotene first into retinol and then into retinol, which is nothing but vitamin A. Okay. However, the rice crop does not really make beta carotene per se, or the enzymes which are present are in very, very low quantities, as a result of which it cannot make beta carotene. So, through genetic engineering, you are going to give the rice crop the ability to be able to synthesize beta carotene. All right. Now, let me just explain what we have this particular pathway in the middle, and the first compound which is listed here is geranyl diphosphate, and this molecule is already present in the young rice endosperm. Okay. You know, if you open the rice, or if you open any other kind of a dal also, for instance, it is a dicotyledon plant, and within the endosperm what you find is that that acts as a storage. So, similarly this particular molecule is already present in the endosperm. So, what does it really lack in good quantity are some of the enzymes which can convert this geranyl diphosphate into beta carotene. Okay. What are these enzymes? The first enzyme which is written here as number 1 is the enzyme phytoene synthase, and it is going to be abbreviated as PSY. Since it is a gene, therefore, it is expressed in the or it is written in italics. So, that is a convention by and large, that whenever when you are trying to write the name of a gene which is normally given a 3-lettered code like PSY stands for a gene which codes for a protein phytoene synthase, all right, then you are going to put it in italics, because then you know it is basically a gene. So, the first enzyme is the phytoene synthase, which converts this substrate into 2-pyrophosphate plus phytoene. The second enzyme, which is numbered as 2, is phytoene desaturase, which converts this into zeta carotene. The same enzyme can also bring about conversion of zeta carotene into lycopene, and finally lycopene can be converted into beta carotene. Okay. And the third enzyme which you see in this pathway is the lycopene cyclase. Okay. So, in other words, when you look at the rice plant or the rice grain in the endosperm, you are basically at this particular stage. Okay. It cannot go all the way up to beta carotene. So, you are going to introduce genes which are going to put these, or they now code for certain enzymes which can bring about the conversion at this stage, at this stage, at this stage, at this stage, and finally accumulate beta carotene. At this stage, once you have synthesized the beta carotene, okay, then the other two enzymes are already present in the rice which can give you vitamin A. So, in other words, now a rice grain, you expect to see that it will also have vitamin A. Okay. So, what does it really achieve? If you have golden rice or the rice which also has vitamin A in it, it is expected to handle the vitamin A deficiency in the developing countries, because otherwise the supply of vitamin A is kind of limited in very poor countries. And similarly, just as I am going to illustrate to you as to how it has been done in the case of rice, this pathway of synthesizing vitamin A has also been introduced into potato, into tomato, into canola, into maize, etc. So, let us see what are the essential steps which are involved. So, here is an outline of the different cloning steps. So, the first thing which we must do is construct an expression cassette which has the required genes, and these are cloned into what is called as a TI plasmid. Now, TI plasmid is a normally occurring is a plasmid in a bacterium, agrobacterium, all right. However, this agrobacterium which contains this TI plasmid, all right, TI also stands for tumor inducing plasmid. Okay. So, this kind of an agrobacterium, which is actually a bacterium which contains this plasmid, has the ability to infect the plants, as a result of which this TI plasmid can now go directly into the plants. Okay. That is what we are going to learn. So, essentially I will repeat this in order to make it clear to you that any kind of a gene, a foreign gene which you would like to introduce into the plant is going to be now first developed into a kind of an expression cassette, and then this entire expression cassette is cloned into a TI plasmid. The plasmid is next introduced into the plants through transformation, and finally you must get a tissue specific expression. Okay. Like what do we mean by that is that eventually the genes which are going to be introduced into the plants, into the rice crop, this gene should be able to get expressed in the endosperm of the rice grain. If it starts to express itself in the leaf or in the roots of a crop plant, that doesn't serve your purpose. What you would like to see is that this vitamin A is going to be made exactly in the rice endosperm. So, in other words, we say that endosperm is a tissue, and what I would like to see is a tissue specific expression of this vitamin A biosynthetic pathway. So, let me explain to you as to how that can be achieved. So, what are the points which need to be considered here? Now, I used the term expression cassette in the last slide. Okay. So, what does an expression cassette means? It means that there is a gene which is going to code for a particular protein. However, this gene is also going to be linked to what is called as a tissue specific promoter. Why? Because as I said that you would like to see expression only in the endosperm. Only then it is going to be exhibited in the rice grain. And number 2, we must have in the expression cassette a transit peptide. And what do we mean by a transit peptide? Now, if you take any kind of a plant cell, okay, this plasmid needs to enter into the plant cells. They also contain a plastid, and plastid gives the characteristic green color to a plant cell, because that is where the chlorophyll is stored in the plastid, which are like small bodies which are inside the plant cell. And so, these plastids, for example, are the sites where photosynthesis is going to occur, and they have chlorophyll. They can also have various kind of pigments like beta carotene and other things. So, in other words, any kind of a foreign protein which you are going to synthesize, okay, must also have a signal in front of that which is going to direct this particular polypeptide into a plastid. Okay. That is where beta carotene is going to be synthesized. And why do we need a transit peptide? It is because the destination of this protein is going to be a plastid. Okay. It should not just remain in the cytoplasm of a plant cell of the endosperm, but rather it should be particularly directed to this particular organelle which is within the plant cell. So, that is what is necessary to understand here. So, let me explain to you as to how do you really make an expression plastid and expression cassette, and what all are the components of an expression cassette. This is the first paper which was on this subject matter, and it was published in the journal Science, and I will briefly explain to you in a very simplified form as to what this plasmid contains. Number 1, what is the plasmid which is used? It is the Ti plasmid. Where does this plasmid normally exist? This plasmid can be introduced into a bacterium which is called as an agrobacterium. This agrobacterium has the capability to infect the plant cell. Okay. So, there are 2 routes which are going to be used here. The plasmid, which has this expression cassette, is going to be introduced into agrobacterium. Then the agrobacterium is going to infect the plant cell, and that is how this expression cassette is going to find itself in the plant cell. So, now let us see what are the important components of the expression cassette. So, what you see here is APH4, this pink region in the plasmid. Okay. So, what does it really code for? It codes for a selectable marker which is called as hygromycin resistance. Okay. Now, the reason for using a selectable marker in molecular biology is that you must have already learnt it in the bacterium, that if the E. coli cell, a simple E. coli cell is going to be transformed with the bacterium, then the plasmid is going to contain an antibiotic marker. So, that now in order to select for those bacteria which have already taken up this plasmid, we can cultivate this bacterium on Lb plus ampicillin. Okay, because you need to distinguish between a bacterium which does not contain the plasmid and the bacterium which contains the plasmid. Okay. So, that is an antibiotic marker. In the same way, when the agrobacterium is going to infect the plant cell, then how do you select for those plant cells which have already got this plasmid inside them? There has to be a selection marker. Okay. And that is what this hygromycin resistance tells you about. that those particular seeds of the plants which will have resistance to hygromycin, they will have acquired this particular plasmid. So, it is used as a selection marker. Okay. And every gene needs a promoter to express. So, this particular gene is expressed under this 35S promoter. This shows you the direction of the transcription. Okay. So, this promoter is actually derived from cauliflower mosaic virus, which is a normal virus which can express itself in the plant cell. So, it is one of the promoters which is used, which is derived actually from a virus, okay, but that gets recognized inside the plant cell. Now, the second thing which you see here is PSY, okay, and this PSY is the phytoene synthase, which is the first enzyme which is required to convert geranyl diphosphate into the first product, which was the phytoene, if you recall that pathway. Okay. So, it is that particular gene PSI, and this is expressed under a promoter which is called as a GT1 promoter, and this promoter is very specific to endosperm. So, in other words, only in the endosperm it gets expressed. The PSY gene can now be transcribed only in the endosperm because this is a tissue specific promoter. Okay. And that is why it needs to be here. So, this is the first enzyme which you are going to be introducing into the plant, and the second enzyme is called abbreviated as CRT1, and this CRT1 codes for phytoene desaturase. Okay, in that particular pathway which I had discussed with you earlier. So, from phytoene, when you go to the next product, you require this particular enzyme which is coded for by CRT1 promoter. Okay. So, this is the kind of that is the reason why we use the term expression cassette, because not only does it have the gene, but it also has the required promoters and it has a selection marker, etc. So, this entire thing will be called as an expression cassette now. In addition to that, what is important here is that just in front of this gene, the PSY gene, you also see here, for example, so there is a leader peptide here. Okay. And this leader peptide, incidentally, the PSY is from a plant daffodil, this particular gene, and this CRT1 gene is taken from Erwinia, which is a bacterium. Okay. Because in genetic engineering, you do have a possibility of introducing genes from any particular source. So, now you have in this expression cassette, so this PSY, that is phytoene synthase, because it has a leader peptide in front of it. Alright, this was already a plant gene from daffodil. So, this allows this particular polypeptide chain, which is going to be synthesized in the cytoplasm, to be directed to the plasmid. So, this gene product or this enzyme is going to be made in the plastid. Okay. Similarly, the second enzyme also has a transit peptide which will direct it to the plasmid. So, both of these enzymes are going to be there. Then you see another marker which is called as kanamycin resistance, because first you are going to put this expression cassette in a bacterium which is agrobacterium. How do you select the transformed bacterium? It is by looking at kanamycin resistance. Okay. So, that is what the function is now of this particular expression cassette. So, now we will see as to what the next steps are. Now, in rice seeds, they have introduced first either only this plasmid, which is PB19 HPC, which contains these two genes PSY and CRT1. Okay. So, that is the gene which is introduced, or into those plants which have already got this plasmid, they have introduced another plasmid which is PZPSC and PZLCYH. So, this particular plasmid, for example, is the same as PB19 HPC, except that it does not contain this hygromycin resistance, and this one provides the last enzyme, which is the lycopene cyclase. Okay. So, in other words, there is a set of plant which already contains this particular expression cassettes. In addition to that, it is transformed with this as well as this plasmid. Okay. Now, what is the process by which we can introduce into plants? So, different steps are explained in this slide. So, what you see here in A is that this is of course not for rice, it is some other seed, but you can do exactly with the rice seeds also. So, the rice seeds are taken, they are sterilized by treating it with some kind of a solution like mercury chloride or something, and now in B what you see is that these seeds are just going to be in the water for some time imbibed, and in figure C you have cultivation of agrobacterium. So, the bacteria which contains this plasmid, which is the TI plasmid containing all the requisite expression cassette, is going to be cultivated in a medium separately. Okay. So, the grains, the rice grain is kept separately here, and the bacteria which contains the plasmid is also cultivated separately in a vessel here. Okay. In the next stage D, what you see, there is infection of the plant tissue with agrobacterium. So, now the seeds here as well as the bacteria are brought together and then they are cultivated for a certain period of time. So that now the agrobacterium is able to infect these seeds. In the next stage, there is a some of the seeds are taken out and they are now plated onto a solid medium. So it is basically an agar plate which contains a filter paper, on top of which these infected seeds are going to be kept. F and G indicates that they are on a special medium which allows these seeds to germinate. So, gradually you see that some plants are coming out, shoots are coming out, the roots are coming out, and what you see here is induction of shoots by certain plant hormones, etc. And in this slide you see the shoot elongation. So, now the seed is germinated, the shoots have elongated, and finally in this medium you see rooting has also occurred. So, both shoots as well as roots are now there in this, and eventually what you have now is that this little mini plant is going to be transplanted. So, in J and K what you see is that once you have a mini plant, and this is only a close up of this part where the roots are growing. So, you see that the roots are also growing here. So, eventually this miniature plant is put into a small pot, it is allowed to grow, and now you see that there is a plant. Now, this is a transgenic crop. Okay. It could be rice, it could be soya bean or whatever, but it contains already all the genes which are required. Okay. So, in a nutshell, I have tried to explain what we saw in the previous slide is that here is an agrobacterium. This is the chromosomal DNA, but this is the TI plasmid which contains your expression cassettes and everything. So, right here what we see is that there is a co-cultivation of the recombinant bacterium and the plant tissue. So, this could be the bacterium and this could be the plant tissue. They are co-cultivated and eventually there is an entire plant which can be developed. Okay. So, endosperm specific promoter will allow this to be produced only in the rice seeds. You also get a tissue specific expression. Now, what is very interesting is that in many of the studies today, what we can do is that you now have a normal rice crop and you also have a transgenic crop. Proteins can be isolated from there, and through electrophoresis these proteins can be separated and we can understand as to what proteins are overexpressed, what are underexpressed. Okay. So, that is what is shown in this slide. For example, this could be the proteins which are isolated from the normal rice crop, and this is the one which produces vitamin A, and all of these dots which you see are really the proteins which can be excised out and they can be studied. So, you know that between a transgenic rice crop and between the normal rice crop, what are the different kinds of proteins which are being produced. And today you have very sophisticated tools which will allow you to do that. And here is a list of some of these proteins. Now, for example, there were proteins related to carbohydrate metabolism, enzymes like pullonases, they are produced 3.3-fold more in the transgenic crop, or UDP glucose pyrophosphorylase is produced 3.46-fold more in the transgenic crop. Okay. So, you really do not stop your experiments at development of a crop, but you can also examine the crop, the physiology and what kind of new kind of proteins that it is making, so on and so forth. Okay. So, it is just all of these things will allow you. So, here is what, for example, is a picture of a transgenic crop, transgenic rice crop. So you see many of these kernels which should have normally been white looking, they look yellow in color because they are making beta carotene. Okay. And if you introduce that last enzyme, which is the lycopene synthase, then they are completely able to convert that into vitamin A, into lycopene, and also finally into vitamin A. Okay. Now, there are only some controversies, because I do not want to leave you thinking that it is all very good for the transgenic crops, and there are some issues which are particularly in Europe and in some of the other South Asian countries. What are these issues? Now, there are many, this could be because of the culture. Now, many people, they would prefer to have white rice. Okay. So, they are not very accepting of this yellow rice. They still want to have the conventional white rice, and they believe that there should be no alteration in God's plan. God's plan was to make white rice. Why are we trying to make it into a yellow rice? So, culturally, there are people who are not very accepting of this. Or it could be because of health reasons, that many people have displayed an allergic response, as a result of which people are very apprehensive, or the level of vitamin A which is presently available in this rice grain is very low. So, people need to consume a large quantity of rice in order to meet the daily requirement of vitamin A, which is not desirable, because they think unnecessarily, why do we have to take a starch containing material. Or it could be because of environmental constraints. Now, between these transgenic crops and the natural varieties, there could be a lot of competition, and the natural rice varieties could just become eliminated, which is not very accepting. Or there could be a threat to biodiversity, contamination of the existing gene pool, or flow of genes from the transgenic crops to normal crops. So, these are some of the things which are still the point of controversy, and people gradually, over a period of time, people will become more accepting of these transgenic crops, but currently there are some hesitations. Now, the final example I am going to illustrate to you is that of Bt cotton. Okay. So, the cotton plants are susceptible to insect attacks, particularly the Lepidoptera insects which are going to attack. And what has been done is that Bacillus thuringiensis is a bacterium which makes a protein which is called as a cryprotein, and this cryprotein is very toxic to this Lepidoptera insects. Okay. So, the idea is that if in the cotton plant itself, if the gene for this cryprotein can be introduced, so that these the cotton crop is able to express this cryprotein, then once this insect is going to attack, it is going to be killed by the cryprotein. So, that is the whole rationale behind development of the Bt cotton. Okay. So, Bacillus thuringiensis cryprotein, which is very toxic, can be introduced into the cotton crop. So, Bt cotton is a genetically modified pest resistant plant cotton variety, and it produces this insecticide. Okay. Now, Lepidoptera insects which I mentioned to you, which attack, these are actually the ball worms, and these are sensitive to this crystalline protein coded for by the cry gene of Bacillus thuringiensis. Okay. So, Bt cotton was accepted, and of course, there were a lot of controversies surrounding that, but by and large it has been planted in the field. So, very briefly, let me explain to you as to what went behind it. Okay. So, again we have an agrobacterium here, which is shown by this orange box, and the bacterium contains its own chromosomal DNA, which is shown, and then it also contains this Ti plasmid. The Ti plasmid contains this cry gene, which can code for this particular toxic protein. Okay. So, now you see that this is a transformed agrobacterium, and it is incubated with this cotton. This is the cell from the cotton plant, and this is the nucleus, and all these are chromosomes inside. Now, after the agrobacterium infection of this particular cell, which is derived from the cotton plant, okay, this particular gene gets integrated into the chromosome. So, now you see that this is a single cell from the cotton plant, okay, and in one of the chromosomes you see this gene which is going to code for the cry protein is now integrated. Okay, it becomes a part of the chromosome, and it can also express itself. This cell is now multiplied in vitro. Okay, cells by putting them in a nutritionally rich environment, they can be multiplied, and then cultivation in vitro the same way that I showed you last time, that you can put phytohormones which will induce shoot, which will induce roots, etc. So, the little plantlets are shown here, and eventually it can be put into a pot like this. So, this becomes now a pest resistant crop. Why? Because this particular cotton plant is capable of making this cry encoded protein, which is going to be very toxic to the insects. So, now let me just summarize whatever we have learned in this lecture, that biotechnology has played a major role in advancement of agricultural practices. We saw several examples, that of biofertilizers, which are written here, transgenic crops, we saw the vitamin A producing rice, the pest resistant crops. All of these are today available only because of our advancement in molecular biology and biotechnology. New areas of science and their applications have been identified, and they should help to increase both the food yield as well as the quality in the coming years. So, I hope with this you get a general idea as to how molecular biology techniques or genetic engineering techniques can be used to introduce new kind of genes into the plants, which will also give very good and beneficial properties to these crops.